Hello student, I am Dr. Chaudhary S.G. from Department of Microbiology, Venkatesh Mahajan Senior College, Usmanabad. I welcome you to my channel Learn Microbiology. In this video, I am presenting a subtopic from BSc third year syllabus, that is the metabolism paper. It is a subtopic of Unit 1st, Enzyme Activation of the enzyme. What is actually mean the activation of enzyme? Enzyme activation is the ability of enzyme that indicates its catalysis nature. What is catalysis? Catalysis is the ability of enzyme that convert substrate into the product. And when it is possible only when the enzyme binds specifically with substrate molecule and convert or modify substrate into the product. It means there must be certain site on the enzyme to bind with the substrate molecule. So in this video we are talking about active sites of the enzyme and its activation. So let us continue. Active sites of enzyme. This is the first point in this video. Actually, what are the active sites? These are the sites that are present on the enzyme. The enzyme molecule is much larger than the substrate molecule. That's why there is a smaller restrictor place which is involved in the substrate binding. So substrate molecule are comparatively much smaller than the enzyme molecule. There exists specific region or the site on the enzyme which serves the binding site for the substrate, and such sites of attachment are called as active sites or catalytic site or substrate site. Here in this figure, the green portion indicates the substrate molecule, the yellow portion indicates the enzyme, and this white portion indicates the groove or the active site of the enzyme. When these active sites occupies the substrate within it and forms ES complex, that is enzyme substrate complex. So due to these active sites, enzyme specifically react to substrate and forms ES complex. Now let us see what are the common features of the active sites. The active site is a small portion of the enzyme molecule with three-dimensional configuration, and it contains specific groups from linear amino acid sequences. So active site having three-dimensional configuration, and it consists of a chain of amino acid with specific groups. For example, ribonuclease enzyme required histidine residue 12 and 119 only for its action. So these residues are necessary for the activity of ribonuclease. Second example, chymotrypsin required histidine residue 57, aspartic acid residue 102, serine residue 195 for its activity or catalytic function. Further, the arrangement of groups in the active site is well defined, which gives marked specificity to the enzymes. These groups or these sequences of the amino acid are specifically arranged. That's why enzymes specifically react with particular substrate. In some cases, there is slight changes in the active site configuration in order to bind with substrate. Though there are changes, the arrangement is specific, no doubt, but sometimes it not happens in all enzymes. In the case of all enzymes, sometimes in certain enzymes there is change in the active site configuration of the enzyme. in order to bind with the substrate according to the substrate binding site. So it indicates the induced fit mechanism of the enzyme action. We will see in detail about the induced fit mechanism of the enzyme action in next video. Here we talk about the active site configuration and activation of the enzyme. Further, the active site binds enzyme to the substrate molecule by relatively weak forces. Means the binding of enzyme molecule with substrate is not firmed. It is unstable at the beginning. So whatever ES complex form is unstable complex due to this weak interactions. The active sites in the enzyme molecule are grooves, services. We observed it in figure also. The white portion forms groove of the enzyme. Okay, so active sites are grooves or services which contains amino acid sequences such as aspartic acid, glutamic acid, lysine, serine, etc. Different amino acid, different groups such as COH, NH2, CS2OH, etc. that serves catalytic group in the active sites. The specificity is due to these groups or amino acids. The services create a microenvironment due to the presence of polar groups, and these polar interactions are essential for the catalysis action of enzyme. Now let us see what actually the enzyme activation is. The enzyme is a biocatalyst. We all know this. It speeds up the rate of reaction without being consumed and retained as it is after the completion of reaction. Means enzyme modifies substrate into the product and freely releases after completion of the reaction without any change. The enzyme particularly acts by binding to the substrate initially, forming enzyme substrate complex, which is transition state of the reaction. So direct conversion of substrate into product is not there. First of all, enzyme specifically binds to the substrate, forms ES complex. This intermediate stage is termed as transition state. The meaning of transition state is intermediate state. Okay, and this intermediate state further, when conformed or become stable, the substrate molecule is converted to product. This is the basic reaction. Transition state is not a stable state. I have already stated this. The transition or intermediate state of formation of ES complex is not a stable state, and it is characterized by free energy, high free energy. The initial ES complex is characterized by high free energy, which is Gibbs free energy, or it is also called as activation energy. So unstable complex is characterized by high activation energy. When further this catalyst particularly acts or proceed the reaction, it lowers the energy of activation. This energy of activation is specifically denoted as EA, capital E, small a. And when this action of enzyme is proceed further, it lowers the activation energy, which is necessary to attain the equilibrium to the reaction and to form a stable transition state. Means when there is decrease in the activation energy following the formation of ES complex, the ES complex becomes stable and the reaction proceed further. So once the stable ES complex forms, it confirms the conversion of substrate into the product. So stable ES complex confirms the formation of products. Let us discuss briefly with the help of graph. In the graph, you are visualizing two peaks. One is in red color and another one is in blue color. The red color peak indicates the reaction without enzyme, and the blue color peak indicates reaction with enzyme. In the case of reaction along with enzyme, the activation energy is very less, while in case of red color peak, the activation energy needed is very high. There are so many reactions in the nature that can be carried out without enzyme, but it takes much more time. It consumes much more time to form transition state complex, and it requires high activation energy. So enzyme catalyst particularly acts by lowering the activation energy and modify substrate into the product. So this is the activation of enzyme, right? Hope you understand the points covered during this video. If you have any doubt, indicate or write in comment section. Thank you for watching.